Good morning to all. Welcome to the Diamond Group of Institutions Neet and K-Cet online classes. Today's topic it is biotechnology applications. So in that biotechnology application, some questions are: first one, Eli Lilly company produces genetically engineered human insulin. Select the correct statement about process and product. So question from the insulin. So insulin, it was produced from the pancreas of Langerhans cells. It can maintain the glucose level in blood. So insulin is a hormone can develop from it is produced from the pancreas of the Langerhans cells. So what's the use of this insulin? Insulin can maintain the sugar levels, otherwise glucose levels in blood. If the insulin concentration was decreased in the body, human beings get the diabetes disease. So now, based upon the question here, the insulin, it was synthesized in the body. It can maintain the sugar levels. If the insulin concentration is not synthesized, means it's not synthesized, the minimum concentration was not there in the body. So the time of the human beings get the diabetes disease. So for the externally, we have to take the insulin into the body, so that artificial insulin will be going to developed by the company of Eli Lilly. So how these are the insulin was developed? So these for the development of the insulin having as a 3 chains. You know those are A, B, C. So A chain, B chain and C chain. A chain having as a 21 amino acids, B chain having as a 30 amino acids, C chain having as a 33 amino acids. So insulin, it will be going to develop from the pancreas in the form of proinsulin. The proinsulin having 84 amino acids. So this proinsulin have converted into the mature insulin. That mature insulin, that is A and B chains are they are connected together by the disulfide bonds. So they are connected together by the disulfide bonds. So this is the A chain and this is the B chain. So now A chain contains as a 21 amino acids and B chain contains as a 30 amino acids. Mature insulins are having 51 amino acids. So the time of production of this artificial insulin by the using of E. coli bacteria done by the that is Eli Lilly company. So based upon that options are, now you see option A, genetically engineered insulin. Human insulin was produced in 1993. Genetically engineered human insulin was developed in the year of 1983. It's not in 1993. And option 2. 2 chains A and B were produced by already joined by disulfide bond. See now, 2 chains A and B were produced by E. coli already joined by disulfide bonds. So here in the E. coli bacteria, the 2 chains will be going to produce separately. So they given as option 2 chains A and B were produced by E. coli already joined by the disulfide bonds. So these are the A and B chains are connected together by the disulfide bonds only. But according to the option they given as they are already producing E. coli bacteria. So basically these A and B chains are artificially we have to synthesize, means we are going to generate it separately in a 2 different types of the E. coli bacteria of plasmids. So these both are not synthesized at a time, means they are not synthesized at the same bacteria in a same plasmid. So option B also incorrect. And coming to the option T, 2 DNA sequences corresponding to A chain and B chain were integrated with the main genome. Peptide chains extracted, joined by disulfide bonds, and human insulin was produced. See now, they given as a 2 DNA sequences corresponding A chain and B chain were integrated with the main genome. Peptide chains extracted, joined, and disulfide bonds and human insulin was produced. By the scene of the third option, A and B chains will be drawn to integrated with the main genome. So integrated with the main genome. Actually, these are the two. These are the two A and B chains will be drawn to integrated with the main genome, not of the human beings. It is integrated with the main genome of E. coli bacteria. So here they given as a main genome of human insulin or human beings. So option C also incorrect. And now we see the option D. Corresponding to A and B chain, two DNAs were integrated in E. coli plasmid. A and B chains was extracted and joined. So basically, generally these are the pro-insulin having as a chain, B chain and C chain. These three will be drawn to separated or that is digested by the restriction endonucleases. They can separate it, they can form as a chain and B chain. This A chain and B chain was integrated into the plasmid of E. coli bacteria. This A chain is integrated into a E. coli of one bacteria and B chain is integrated into a E. coli of another one of the bacteria. Like that, this A and B chains are multiplied or cultured in A and B chains at different E. coli bacteria. Later, these both are combined together. These two chains are selected and extracted. In between the A and B chains, they can form into the disulfide bond. So now this insulin we call as a mature insulin. So answer is correct. Coming to the next one, match the column 1 with column 2 and choose the correct combinations from given below. See here the column 1, column 2 they given as column 1 A lepidopterans, B BT toxins, C coleopterans and D diphterans. Column 2, 1 proteins, beetles, flies and armyworms. You know that biotechnology will be going to use into the different sectors in plants and animals, that is in agriculture and medicines. So by seeing of the agriculture biotechnology, that is biotechnology applications in agriculture. So we know about the concept of BT totem. So at the time of BT toxins will be grown extracted from the bacteria, will be grown to control the different types of the insects. Some of the different types of the insects are. So they belongs into the genus of that is some of the insects, that is they belongs into the genus of lepidopterans. Under the genus of lepidopterans will be grown to use as armyworms and tobacco budworms. So these are the two types of the insect, that is armyworms and the tobacco budworms will be known to controlled by the BT toxins. And in the same way, coleopterans. So these are the coleopterans. Under the coleopteran they are having some that is beetles. Beetles are they belongs into the coleopterans, and diphterans they belongs into flies and mosquitoes. So you know these are the BT toxins, Bacillus thuringiensis. Bacteria can produce a specialized toxins. Those toxins are they are present in the form of proteins. So those proteins are we call as a crystalline proteins. So now lepidopterans, under the lepidopterans genus, armyworms and tobacco budworms, and BT toxins are the specialized proteins will be known to develop from the Bacillus thuringiensis bacteria. Coleopterans, under the coleopterans beetles are included. Diphterans, under the diphterans flies and mosquitoes are that is included. So by the matching of these lepidopterans match with the armyworms, BT toxins match with the proteins, coleopterans match with the beetles, and the diphterans match with the flies. So answer will be going to get into B. That's next question. The given flowchart summarizes the event involved in making tobacco plant resistant to nematode meloidigin incarnation. The question from the RNA interference concept. So many different types of the nematodes are the causes diseases into plants and animals and human beings. Like One of the nematode has meloidigin internisha. It can process the disease in a tobacco plant. So that meloidigin internisha, it was present in a soil. It can enter into the tobacco plant roots. So by the entering of this meloidigin internisha into the tobacco plant cells or tobacco plant, so those are the roots of tobacco plants will be going to damage. When the roots of the tobacco plants are damaged, the yield automatically will be going to decrease. The concept of this flow chart, it belongs into the RNA interference concept only. So we have to reduce the infection of meloidigin internisha nematode into the tobacco plant. So we can use, as by the using of biotechnology, we can control the infection of meloidigin internisha in tobacco plants. Further, we have to collect the nematode specific gene. So we are going to isolate the nematode specific gene. Nematode specific gene. That nematode specific gene will be going to present as a double-stranded DNA. That double-stranded DNA it was synthesized by the process of reverse transcriptase mechanism, and here this double-stranded DNA having two strands. One of the strand it is sense strand, another strand it is antisense strand. So you know about the out of the two strands of DNA, one strand of the DNA only can form to the mRNA. So but what is the DNA will be going to develop or created by the process of reverse transcriptase mechanism. The two strands of the DNA can produce into the mRNA. So that is a major concept, or that is a major thing of this reverse transcriptase process mechanism what the double-stranded DNA will be going to develop. And that DNA we call as a nematode specific gene. That nematode specific gene we have to introduce into the plant cell. So you can imagine this is the plant cell. So here the nematode specific gene is there. So this is the double-stranded DNA. So this is only we call as a nematode specific gene. So it can produce both RNAs of two strands, sense and antisense strand. Means double-stranded RNA will be going to formed from these nematode specific gene. So the nematode will be going to enter from the soil into the plant root cells. So that nematode also having double-stranded DNA. So now this double-stranded DNA means nematode gene is. So it can produce the mRNA by the process of transcription. So whenever these are the mRNA, it was developed from the nematode gene. In the same time, at the same time, that nematode specific gene we are going to introduce into the plant cell, it is also then produced into the double-stranded RNA. So after the formation of this double-stranded RNA from the nematode specific gene, don't get confused here. Nematode specific gene can produce the double-stranded RNA, and the nematode gene can produce the single-stranded RNA. So the concept we are going to discuss in now this double-stranded RNA is developed from the nematode specific gene. After the formation of double-stranded RNA, by the involvement of RNase enzyme, by the involvement of RNase enzyme and RISC enzyme. RNase enzyme and RISC enzyme. The RISC enzyme name it is RISC, abbreviation is RNA induced silencing complexes. It can cleave the double-stranded DNA into small pieces. This double-stranded DNA will be going to cleaved by the small pieces by the activity of RNase and the RISC, RNA induced silencing complex. So now after the treating of this RISC enzyme, dicer enzyme will not treated into the double-stranded RNA into many number of small double-stranded RNAs. So now these are the dicer enzyme will be going to work after the formation of small double-stranded RNAs. Here the RISC enzyme, RNA induced silencing complex, the double-stranded RNA will be going to here. The small fragments of the double-stranded RNAs are separated and formed into small single-stranded RNAs. So now here the single stranded RNAs are both will be going to having sense and antisense. These both are. This is the same antisense strand and this is you can imagine as a sense strand. So now here these are the RNA molecules, that is sense and antisense RNA molecules. Those are complementary to the RNA of nematode gene. What's the mRNA? It was developed by the process of transcription. So this is complementary to the RNA of the nematode gene. So now here these are the antisense RNA will be going to come and bind to the nematode gene mRNA. They can form into double-stranded RNA. If the double-stranded RNA is formed, so by the formation of double-stranded RNA, we know about the RNA interference concept. Otherwise, mRNA silencing. Expression of the nematode gene will be going to prevented in the form of protein. If the protein is synthesized by the nematode of memorizing interstitia, the tobacco plant will be going to get the disease and yield automatically reduced. So to prevent the expression of nematode specific gene in the form of a protein, we have to synthesize the complementary RNA molecule. That complementary RNA molecule will be going to developed by the nematode specific gene. So that nematode specific gene can produce the RNAs. That these RNAs will be present in the form of double-stranded RNA. Means here the nematode specific gene, double-stranded DNA can produce the double-stranded RNA. Means sense strand and antisense strand both will be going to produce the RNA molecule. They can form into double-stranded RNA. And these double-stranded RNA is by the involvement of dicer enzyme will be going to split into the main number of fragments. After the formation of small number of fragments, RISC enzyme will be going to used to the conversion of these double-stranded RNA molecules into small single-stranded RNA molecules. Out of these single-stranded RNA molecules, RNA molecule will be going to bind into the nematode gene RNA, nematode gene mRNA. So now they can form into a double-stranded RNA or double-stranded mRNA. If the double-stranded mRNA is formed, mRNA of the nematode gene is silenced by the formation of double-stranded RNA. So this is also called as a mRNA silencing. This is called as a mRNA silencing or also called as a RNA interference. Here the protein synthesis not takes place or protein synthesis stops. If the protein synthesis stops, the nematode will not get the food materials, later it will be going to die. Like the way we are going to prevent or we can perform the RNA interference by the using of biotechnology. So based upon the concept only, they are given as a flow chart. You know, double-stranded DNA produce approximately 21 to 23 nucleotide RNA molecules. So now these double-stranded this double-stranded DNA will going to produce into the small fragments of double-stranded RNA molecules. So that size will be going to present in the form of 21 to 23 nucleotide RNA molecules. And later, now RNase enzyme dissect up the double-stranded RNA molecules into small interference RNA molecules. So now here the double-stranded RNA is cleaved by the involvement of dicer enzyme can form into a small double-stranded RNA molecules. These are only we call as siRNA, small interference RNA. So after the formation of the small interference RNA, here the enzyme of RISC will be going to involve and they can form into the single-stranded RNA molecules. See now here the small interference RNA molecules will be going to activated. So by the formation of the small interference RNA molecules, by the activation of siRNA, RISC enzyme will be going to activated. By the activation of RISC enzyme, the small siRNA is converted into single-stranded RNA molecules. Single-stranded RNA molecules. After the formation of single-stranded RNA molecules, if these small single-stranded RNA molecules are formed, they contain as a sense and antisense strand. One of the antisense strand will be going to directly bind on the surface of the nematode gene mRNA. They can form into a double-stranded RNA molecule. They can prevent the protein synthesis. So this is it. So based upon this, RISC cut the transcript area where siRNA binds into the miRNA. See now, these are the siRNA will bind into the mRNA, and mRNA parasite is destroyed. Means after the formation of double-stranded mRNA of nematode gene, they can prevent the synthesis of protein. They can stop the synthesis of protein. So now that nematode will be going to die. So that is a concept. You know these things you will be get easily. So based upon the concept, options are identify the steps X and Y in the given flowchart. So now X means here and Y means here. In these two steps of the X and Y, what will be going to happen? So you know directly in the stage of the X, after the formation of double-stranded RNA molecules. So after the cutting of the double-stranded RNA molecules in the form of a small by the using of dicer enzyme. So now these are the double-stranded RNA molecules will be going to undergoing by the activation of RISC enzyme. RISC enzyme will be going to activated. After the activation of the RISC enzyme, here the RISC enzyme will be going to activated. When the RISC enzyme is activated, the double-stranded RNA molecules are then converted into a single-stranded RNA molecules. So now, see now, the siRNA unwinds the RISC is activated. Here the RISC is activated. Whenever the RISC enzyme is activated, that small induced RNA will be going converted into the small single-stranded RNA molecules. Previously it was having as double-stranded RNA, but it is converted into a single-stranded RNA molecules. When the single-stranded RNA molecules are formed, they can bind directly into the mRNA of nematode gene. See now, RISC here. These are the single-stranded RNA molecules will be going to formed by the presence of RISC enzyme. Now, RISC cut the transcript areas where siRNAs bind into the mRNA. So it's here only these single-stranded RNA molecules are formed directly. They can bind into the RNA of nematode gene. They can prevent the protein synthesis. So here, option Y, the activated RISC targets complementarity mRNA molecules. Whenever the RISC enzyme is formed, single-stranded RNA molecules are formed, they directly bind into the mRNA of nematode gene. And option D, siRNA complexes with deoxyribonuclease to form as a RNA. Here, they can given as siRNA complexes with deoxyribonuclease enzyme. This is not a deoxyribonuclease enzyme, it is a RNase enzyme. So option B is incorrect. And option C, the activated RISC targets complementary mRNA molecules. The activated RISC targets complementary mRNA molecules. Here, they given as a in the place of X according to the C option. Here, they given as a Y, and in the place of Y, they given as a X. Actually, it should be present in the form of reverse order. So, firstly, siRNA complexes with the ribonuclease to form as a RNA-induced silencing complex. Means here Y should be present in the place of here, but they given as a first position respectively in the form of a X and Y. So option C is incorrect. And option D, the deactivated RISC targets the complementary tRNA. It's not a tRNA, it's a mRNA. So according to the correct answer, it is in the place of X, small induced RNA complexes with ribonuclease enzyme, and in the place of Y, the activated RISC targets the complementary mRNA molecules. It says as here, after the formation of double-stranded RNA molecules, dicer enzyme can cleave and they can form into the small double-stranded RNA molecule. Later, RISC enzyme is activated. That RISC will be going to cleave the double-stranded RNA molecules into small interference RNA molecules. That small interferon RNA molecules directly bind into the mRNA of nematode gene. They can form into the double-stranded RNA. They can prevent the synthesis of protein by the formation of double-stranded mRNA. Protein synthesis will be going to stop. That is, translation mechanism is stopped. Here the mRNA will be going to inactivated or silenced. So that's why this is called as RNA interference or mRNA silence. So answer it is A. Next question. Fruit softening enzyme in tomato is. So tomato fruit softening enzyme, it is a polygalactourinase. Actually, you want to know the applications in biotechnology. The biotechnology applications, by the using of the biotechnology, we can reduce the ripening of fruit. Tomato, the new variety of the tomato, it is flavor sour tomato. In these are the flavor sour tomato, ripening of the tomato will be going to delay. So here we can see as a tomato fruit cell wall. At the time of ripening, this polygalactourinase enzyme is activated. If the polygalactourinase enzyme is activated, the cell wall will be going to degrade and it will be going to form as the fruit will be going to become as a smooth. So to reduce the, otherwise to prevent the activity of polygalacturonase enzyme. By using our biotechnology, we can prevent the activity of polygalacturonase enzyme automatically. So those are the tomato fruit ripening will be going to delay. So now they divide as an options here: zymase, enterokinase, plant starboard slave. Zymase enzyme we are going to see into the alcohol production, and the enterokinase enzymes and the plant starboard slave enzymes are we are seeing into the disrupt cycle and remain as a polygalacturonase enzyme. This polygalacturonase enzyme we are going to use into the ripening of tomato. So this ripening of the tomato fruit will be going to done by the polygalacturonase. If the polygalacturonase enzyme is activated, tomato fruit will be done ripen fastly. If the polygalacturonase enzyme is prevented, so the tomato fruit ripening will be going to delay. To delaying the ripening fruit of tomato will be going to prevent the activity of the polygalacturonase enzyme. So answer is D. And question number 5. Match the column 1 with the column 2 and choose the correct combination from the option given below. Column 1 and column 2. Column 1 they given as RDA technology. Toxins of BT is activity. BT is activated by ELISA is based on, and option D gene therapy. Column 2. Alkaline pH of blood. ADA deficiency. Non-conventional method of diagnosis. Antigen antibody interactions. See now recombinant DNA technology. So by the process of recombinant DNA technology, we can create the new variety of the organism by combining the two different types of the genes, two different types of characters. So options are here: alkaline pH of blood, ADA deficiency, non-conventional method of diagnosis, and antigen antibody interactions. So now the recombinant DNA technology, by using of the recombinant DNA technology, it is a non-conventional method of diagnosis. So here the PCR technique, ELISA technique, we are going to use into the detecting of diseases. These all belongs into the PCR and ELISA techniques are related steps of recombinant DNA technology. So DNA is a non-conventional method of diagnosis. And option B, toxins of BT is activated. You know the BT toxins or Bacillus thuringiensis genes will be going to extracted from the Bacillus thuringiensis bacteria. So for the cell production, these bacteria can produce the BT toxins. The BT toxins will be going to present in inactive condition whenever the organism will be going to. Otherwise, when the genes or toxins will be going to present as acidic pH, otherwise we call as a alkaline pH. The alkaline pH will be going to present in a intestine of insect intestine. So if the insects are taken these BT toxins, automatically the inactive toxins will be going to activated and the midrod of the insect will be going to swell. Later it will be going to burst. Insect will be going to die. That's the concept of BT toxins. So toxins are activated by the alkaline pH of blood. And ELISA based on enzyme linked immunosorbent assay it is. So at the time of diagnosis of the diseases in early conditions, we are going to using as this ELISA test. The ELISA test is based upon the antigen and antibody interactions. And last one is gene therapy, removing of the faulty genes from the organism and adding of the functional genes. That's called as a gene therapy. So for the gene therapy, is example as one of the disease as SCID, server combined immune deficiency disease. It is occurred by the ADA deficiency, adenosine deaminase enzyme is deficiency. So by the process of gene therapy, we can remove or we can solve the ADA deficiency. So based upon this answer is A, that is RNA technology. It's a non-conventional method. A is 3. So out of the given options, A is 3 is the only, so B is answer. And next question, BT toxin gene has been cloned from the bacteria and expressed in plants, provide resistance to insects without the need for insecticides. Examples such plants. See now, BT toxin genes as cloned from the bacteria. So now they can express in a plants. They will be giving a resistance into the insects without using of the insecticides. So those are the insecticides. We are no need to use our insecticide chemicals. Without using other insecticides, we can prevent the insects. So such type of the plant examples are, those are option they given as cotton and corn, rice and potato, tomato and soybean, and answer it B is all of this. Answer it is all of this because what are the plants are they given in the question of option A and B and C. Those all of the plants we are going, they belongs into Bt varieties. Here the Bt varieties represent as Bt means Bacillus thuringiensis. This Bacillus thuringiensis genes will be not extracted from the bacteria of Bacillus thuringiensis. They can produce the Bt genes or Bt toxins. So these all are the plants will be going to create they will going to be a resistance without adding of insecticides. So answer it is all of this.